Hello, my name is Anika Liu. I'm a research scientist at Boehringer Ingelheim in GCBDS, which stands for Global Computational Biology and Digital Sciences. The work I'm presenting here is part of our efforts in indication expansion, which aims at finding new therapeutic applications for drugs or drug targets and is therefore quite similar to drug repurposing. So in GCBDS, this is the aim here is to enable and accelerate this process using data-driven solutions, and for that we're using a wide range of data sources and methods to generate different evidence streams, including genomics, transcriptomics, and text mining, for example. Thereby, of course, each data stream has its own strengths and limitations, which is why the final suggestions are based on the combined evidence and additional sanity checks. So in this poster, I'm presenting one of the sanity checks we've been working on, namely filtering out indications which are proposed for a certain target, although the target itself is not expressed in disease-relevant tissues. This is done in two steps, which is here shown on the right. In the first step, we identify disease tissue links, and then we query target expression in the disease-related tissues. While the Human Protein Atlas was a straightforward choice as first resource for baseline expression data, finding a good resource for disease tissue links was not as easy. Here we found that the coverage of diseases was not great often, and also the focus was often set on symptomatic tissues, while also others may be relevant mechanistically. So for our use case, covering more disease tissue links was essential, while we could tolerate a few false positives, and therefore we looked at the co-occurrence of disease terms and tissue terms in Medline abstracts, and whether this is enriched in comparison to the overall frequency of disease and tissue mentions, respectively. So how well does this process work? For that, we looked into how well we can recover known gene disease associations from metabase. Here across all gene disease pairs, 87% showed expression in disease-related tissues, which is more than if you'd look at a random combination of genes and diseases from metabase, where only 82% showed expression. So this is a small enrichment. However, we'd expect to have a higher percentage with expression close to 100%. Therefore, we first looked whether this percentage depends on the confidence of associations and metabase, and indeed found that it increases to 97%. So here, this part of the figure would be that for highly confident interactions. So for the associations with lower confidence, so here on the left side, the lower percentage may not only be a limitation of the approach, but also partially due to false positives. So to better understand where the process did not work, we then looked into which diseases and targets were most frequently missed in absolute terms and missed in at least half of the time in relative terms. So on the disease side, the most frequently missed one is mouse diseases, and there are a few more shown in the figure here. So these don't have much in common from the biological perspective, but they are found because of the same limitations, namely the fact that we didn't include the relevant target expression tissue disease links, either because there wasn't a clear tissue to link the disease to, or because expression was not available for it. So here what we can improve on is including more baseline expression data beyond the Human Protein Atlas. But for some cases, this also wouldn't be enough, for example, in case of immune-mediated diseases, which are not clearly linked to a specific tissue. We then looked into the most frequently missed targets, and here found an enrichment of proteins linked to inflammation and immune response, and especially extracellular ligands. As immune cells are not primarily tissue-based, this explains why they are missed, and this may even be beneficial because these proteins are often rather biomarkers than targets, but this remains to be explored. Furthermore, genes which are only induced in diseases are missed, so triaging should not be applied to this type of target. So in summary, text mining based disease tissue links can aid in the evaluation of target expression in disease-relevant tissues in our approach. Overall expression in disease-related tissues is enriched, especially among highly confident gene disease links, indicating that this can help to identify true links. However, the approach is limited to tissue-based mechanisms of action with matched expression data. Hence, including baseline expression data from additional tissues can improve the performance, but this also remains insufficient for some diseases and targets. As introduced already in the beginning, this work is only a small part of our indication expansion efforts at Boehringer Ingelheim, and I'd therefore also like to highlight a poster on knowledge graphs, which we also use for indication expansion and which is part of the Boehringer Ingelheim-Yale collaboration. Last but not least, thanks to you for stopping by at this poster. Feel free to reach out with questions and comments, and have a great time at ISMB.